So we have already discussed about the globular proteins and the fibrous proteins. We said that globular proteins they have the spherical shape or the spherical contour or the spherical shape, whereas the globular fibrous proteins have the extended structure, whereas the globular proteins have the spherical three-dimensional structure. And I have already highlighted that the fibrous proteins have mainly the structural role, like as we say collagen, keratin, and elastin. All these are fibrous proteins. They have the structural role. Coming to the globular proteins, they have many functions like storage, transport, catalyst, defense mechanism, many, because most of the globular proteins have mainly catalytic function or transport or storage and so on. And since the fibrous proteins, they have the extended, elongated structure, they are mostly insoluble in water, whereas globular proteins are soluble in water. And these fibrous proteins have the repeated amino acid sequence. This you will see when we discuss about the structure of the collagen. In collagen, every third amino acid is the collagen structure, every third amino acid is a glycine. That time we will elaborate. And of course, the globular proteins have irregular amino acid sequence. And the fibrous proteins, they are less sensitive to the changes in heat and the pH, whereas these globular proteins are sensitive to the changes in heat and pH. As I already said, that these globular proteins loses its three-dimensional structure with either alcohol or the heat or the when we change the pH, because the hydrogen bonds and other bonds would be the non-covalent bonds that are stabilizing the three-dimensional structure would be disrupted. As a result, the protein loses its three-dimensional shape. And as we already said that most of the proteins are globular proteins. And coming to this one, as we know that globular proteins have the globular structure, and according to your syllabus, we need to study the structure and function of the hemoglobin, myoglobin. These are globular proteins. Then collagen, keratin, and the silk fibrin. So these two globular proteins and these fibrous proteins we will be discussing in this module. And so now we will discuss elaborately about each and every protein structure and its function and in detail. So we will start with the hemoglobin, and we know that hemoglobin is a protein that is mainly responsible for carrying the oxygen from lungs to tissues. And we know that the hemoglobin, red color of the hemoglobin blood is because of the red color of the blood is because of the hemoglobin present in the red blood cells. And this hemoglobin is present in the red blood cells. You can see here, this is the red blood cell, and the red blood cell, the hemoglobin is present inside the red blood cell. That's why it is intracellular protein. That means it is present inside the red blood cell. So that means it is intracellular protein or the cytoplasmic protein. And each red blood cell has many hemoglobin molecules. You can see here roughly 270 million hemoglobin molecules present in each red blood cell. So that means the red blood cell, the 90% of the weight of the red blood cell is constituted with only hemoglobin molecule. That means the red blood cell has only hemoglobin. That is a major component present in the red blood cell. That means 90% of its weight constitutes with the red blood cell. And we know that this hemoglobin has is a tetramer, in the sense it is a multimeric protein. It has two alpha chains and the two beta chains present, and all these are associated with each other for exhibiting its function. And this is about the overview. And as I already said, that hemoglobin main function is to carry oxygen from lungs to tissues and uh, carry back carbon dioxide and protons to the lungs from the tissues. And we know that if you take in adult humans have roughly 20 to 30 trillion red blood cells at any given time. So that means 90% of the weight of the red blood cell is constituted with the hemoglobin, and each red blood cell has 270 million hemoglobin molecules, and adult humans are roughly 20 to 30 trillion red blood cells at any given time. And coming to the details of the hemoglobin, hemoglobin has three important functions and three important properties. Three important functions and three important properties. We know the important functions, that is, it's the three important functions are: one is it is it has the ability to transport and oxygen from lungs to tissues. That means it transport oxygen from lungs to tissues. And the second one is it also transports carbon dioxide from tissues to lungs. And third one, it is also involved in the regulation of pH of the blood. So these are the three important functions. That means O2 transport and the carbon dioxide transport and regulation of the pH of the blood. These are the three important functions of the hemoglobin. And coming to the three important properties, and three important properties. Hemoglobin is good in oxygen binding. So it is good in oxygen binding, and it is also good in O2 releasing, oxygen releasing, where it needs to release and when it needs to release. So as I already said, that the main function of the hemoglobin is to carry oxygen from the region of the lungs to the tissues. So in the region of the lungs, the partial pressure of oxygen is very high, that is 100 torr, whereas the region of the tissues, the partial pressure of oxygen is only 20 torr. So hemoglobin, when it moves to the lungs, it is loaded with the oxygen, then it becomes oxyhemoglobin, and this oxyhemoglobin would travel in the circulation, then near the tissues, and the hemoglobin release its bound oxygen. So that is the it is the main function. And since hemoglobin is good in oxygen binding, it is also good in oxygen releasing, whereas that's why the in evolution the hemoglobin is chosen to transport oxygen instead of the myoglobin. Myoglobin is only oxygen storage protein, so that you will understand later. Because myoglobin is also good in oxygen binding, but it is not good in oxygen releasing where it needs to release. So that's why the hemoglobin is good in oxygen binding and is also good in oxygen releasing. And the third important property is the hemo. Since it is a multimeric protein, it exhibits cooperativity. Cooperativity in the sense, so binding of one O2 molecule to the one subunit. Brings a conformational change. As a result, hemoglobin affinity towards the other, oxygen affinity towards the other subunit, as other subunits increases. So that means, as I already said, that hemoglobin is a tetramer. It has two alpha chains and two beta chains, and so each chain has one O2 binding site. So that means binding of one oxygen molecule to the one subunit brings a conformational change, and which in turn increases the O2 affinity towards the other subunit. So this phenomenon is known as the cooperativity. That means binding of one ligand makes the further ligand binding more easy and more likely. So this is exhibited by the multimeric proteins. Here the hemoglobin is also multimeric protein, and it exhibits the cooperativity. You can also see in this diagram that it is a tetramer, and you can see here with increasing binding of first oxygen molecule to the first subunit, first oxygen molecule first oxygen molecule to the first subunit increases hemoglobin affinity towards the second oxygen molecule. Binding of second oxygen molecule to the second subunit increases its affinity more towards the third oxygen molecule. Binding of third oxygen molecule increases the hemoglobin affinity towards the fourth oxygen molecule. So you can also clearly see here that that means one ligand binding makes the further ligand binding more easily and more likely, and so this phenomenon is known as the cooperativity or the allosteric behavior. So this not only hemoglobin, many multimeric proteins exhibit this property, that is the cooperativity. And here not only binding, so losing of one oxygen molecule also makes the further oxygen molecules can easily lose from the hemoglobin O2 binding sites. That means not only it is binding, it is that's why it is also good. It is also good in releasing where it needs to release. And so this is about the three important functions, three important properties. Three important functions is it transports the oxygen from lungs to tissues, and it transports carbon dioxide from tissues to lungs, and it is involved in the regulation of the pH. And second, three important properties: it is good in oxygen binding, and it is also good in oxygen releasing, or where and when it needs to release. And third, it exhibits the cooperativity. And these are the three important functions and three important properties. Then coming to so so this is about the overview. Coming to the details of the how the hemoglobin is able to carry the oxygen and what is its structure. So to understand this, we need to understand the detailed structure of the hemoglobin. So in a hemoglobin, as the name itself says, that hemoglobin has a heme part as well as the as well as the as well as the globin part, heme part as well as the globin part. So we need to heme part is the that's where the O2 binding takes place. Globin part is the protein part. This is the protein part. It is the non-protein part of the hemoglobin. And we will discuss one by one. First, we will discuss about the heme part, and second, then we will discuss about the globin part. And you can see here that the hemoglobin is a tetramer, as I said that you can see the structure of the heme here. This is the hemoglobin, and it is a tetramer. It has two alpha chains and the two beta chains, and each chain has each subunit has the heme pocket. That's where the oxygen binding takes place. This is the structure of the hemoglobin, and for the sake of to make you a little more clear, I will just draw this structure here such that you will able to understand. So if you see the structure of the, as I already said that hemoglobin has the component as well as the globin component. This heme component is the non-protein part of the hemoglobin, and this heme component has organic component. Second one, iron atom. So that means it has organic component as well as the inorganic component. And the organic component is nothing but the protoporphyrin system, protoporphyrin ring system, whereas the inorganic component is the iron atom that is present in Fe plus 2 state. So now the coming to the protoporphyrin system. This protoporphyrin system, you can see here, it has I will just draw, and later before that you can see here, this is the protoporphyrin system, and also iron is present in the center. And if you can see carefully this structure, it has many components, and the simplest way to remember and draw this structure is that this protoporphyrin is composed of 4 pyrrole rings. It has four pyrrole rings. So we know pyrrole, this is the structure of the 4 pyrrole rings, and these 4 pyrrole rings are, let me draw simultaneously. So 4 pyrrole rings, you can just see here that this is the one pyrrole ring, and here this is another pyrrole ring. This is only the cartoon diagram. Third pyrrole ring. This is fourth pyrrole ring. So that means it has four pyrrole rings, and of course we have not placed double bonds. This we will place it later. And these 4 pyrrole rings are linked by 4 methylidine bridges. So you can see here, these 4 pyrrole rings are linked by 4 methylidine bridges. Here, that means here it is CH2, 4 methylidine bridges, and here it is also again linked by one more methylidine bridge, one more methylidine bridge, one more methylidine bridge. So 4 methylidine bridges. Then in addition to this, it has 4 methyl groups, 4 methylidine bridges, and 4 methyl groups. And you can see here the one methyl group is here, CH3, and one methyl group is here, CH3, and another methyl group is here, CH3, and one more methyl group is here, CH3. 1, 2, 3, 4, for the sake of understanding. So 4 methyl groups, and then 2 vinyl groups, CH double bond CH, 2 vinyl groups. 2 vinyl groups are also present, and one vinyl group is here. This is the one vinyl group, and this is the another groups, 2 vinyl groups, then 2 propionic acid chains, 2 propionic acid chains. So one propionic acid chain is here. So 1, 2, 3. One propionic acid chain is here, and other propionic acid chain is here. 1, 2, 3. These are the propionic acid chains. These are the 2 propionic acid chains. And now the iron atom is present in the center. It is present in the Fe plus 2 state, and in Fe plus 2 state, here this can form 6 coordination bonds. 4 valencies are satisfied by the 4 nitrogen atoms of the pyrrole ring, 1, 2, 3, 4, and the 5th valency is satisfied by the histidine atom of the histidine nitrogen of the histidine imidazole nitrogen of the histidine that is part of the globin chain. So that means this is the 5th coordination site, and the 6th coordination site. This is the 6th coordination site. This is meant for O2 binding. And so now since it is a 4 pyrrole rings, it is composed of 4 pyrrole rings, and these 4 pyrrole rings are connected by the 4 methylidine bridges, and the 4 methyl groups are present, 2 vinyl groups, 2 propionic acid chains. Iron atom is present in the center, and it is present in the Fe plus 2 state. In Fe plus 2 state, it can form 6 coordination bonds, and 4 valencies are occupied by the 4 nitrogen atoms of the pyrrole rings, and the 5th valency is satisfied by the histidine nitrogen of the globin chain, and the 6th oxidation site is meant for the O2 binding. So now, since it is a 4 pyrin, we have not placed the double bond. You can see here double bond and a single bond, again double bond, single bond, again double bond, again single bond, double bond, again single bond, again double bond, again double bond here, again double bond. So again double bond, single bond, again double bond, single bond, again double bond, single bond, again double bond. So this is the structure of the heme. So you can understand by you can see when you look at here, you may not able to understand very quickly. So when you split this molecule into individual components, you will understand that it has 4 pyrrole rings, 2, 1, 2, 3, 4 pyrrole rings, 4 methyl groups, 1, 2, 3, 4, 2 vinyl groups, 1, 2, and 2 propionic acid chains, 1, 2, and the iron atom is present in the center, and it is present in the Fe plus 2 state, and in Fe plus 2 state it can form 6 coordination bonds, and 4 valencies are occupied by the 4 nitrogen atoms of the pyrrole ring, and the 5th valency is occupied by the histidine of the protein component of the hemoglobin, that is globin, and 6th valency is meant for the oxygen binding. So that means you see here that oxygen is binding in the at the iron that is present in the center of the heme. So this is the structure of the heme. You can also see here And when you look at the structure, this is the each subunit. Each subunit has one heme pocket, and each heme pocket has the heme structure, and the iron atom is present in the center. That's where the oxygen binding takes place. So this is the structure of the heme. Coming to the this is the structure of the heme. So coming to the globin. So globin part of the hemoglobin. Coming to the globin part of the hemoglobin, as I already said that hemoglobin is a tetramer. That means it has four subunits. It is a tetramer. It has four subunits are the four polypeptide chains, and these are two alpha chains and two beta chains. It has two alpha subunits and two beta subunits, and the alpha subunit has 141 amino acids, whereas the beta subunit has 146 amino acids. Alpha subunit has 141 amino acids, beta subunit has 146 amino acids. Therefore, since it is a tetramer, it has two alpha subunits, means 141 into 2, that is equal to 282 amino acids, and two beta subunits 146 into 2, that is equal to 292 amino acids. Total, it has 574 amino acid residues in the globin part of the hemoglobin molecule. And so coming to the alpha subunit, in the alpha subunit, the N-terminal amino acid in the alpha subunit, the amino terminal amino acid. This is the amino terminal of the polypeptide chain, and this is the carboxy terminal of the polypeptide chain. The N-terminal amino acid is valine. The N-terminal amino acid is valine, whereas in case of the C-terminal, it is arginine. And whereas in case of beta subunit, the amino terminal amino acid is again valine. Amino terminal amino acid is again valine, and whereas the carboxy terminal amino acid is histidine. Carboxy terminal amino acid is histidine. So since the hemoglobin is a tetramer, and so if you write the cartoon diagram of this one, and let me draw once again. Cartoon diagram, you can just see here, since it is a tetramer, hemoglobin is a tetramer, it has four subunits, and so this is the cartoon diagram. This is the one subunit, and this is the protein part of the, this is the one subunit, and here this is the another subunit. This is the third subunit. This is the fourth subunit. You can call it as either alpha 1, alpha 2, or beta 1, or beta 2. So each subunit, so this is the globin part, and each subunit has one heme pocket. Heme pocket is present here. Heme pocket, as I already said, this is the non-protein heme pocket. That's where in the heme pocket has the iron Fe. It is present in the Fe +2 state, so that each hemoglobin has 4 subunits, and with 4 heme pockets, Fe +2. So that means it is a tetramer, and it has 4 subunits, and it also has 4 heme pockets. So that means that's where the oxygen binding takes place. This is the site where the oxygen binding takes place. So that means this hemoglobin has heme part as well as the globin part, and the heme part is the non-protein component, whereas the globin part is the protein component. And the heme part has a heme part is made up of with the protoporphyrin ring system, and it has a 4 pyrrole rings, 4 methylidine bridges, 4 methyl groups, 2 vinyl groups, 2 propionic acid chains. And the center of the 4 pyrrole ring system, the iron is present in its Fe, in its +2 oxidation state. In that it can form 6 coordination bonds, and 4 valencies are satisfied by the 4 nitrogen atoms of the pyrrole ring, and the 5th valency is occupied by the imidazole nitrogen atom of the histidine that is present in the globin chain, that is the protein component, protein part, and the 6th valency is meant for the oxygen binding. So that means in a deoxygenated state, it is empty. In oxygenated state, oxygen binds here. And coming to the globin part, it is a protein part of the hemoglobin, and it has 4 subunits, and that means 2 alpha subunits and the 2 beta subunits. And alpha subunit has 141 amino acid, beta subunit has 146 amino acid, and therefore it has a total 574 amino acid residues. In both the subunits, alpha and beta, the amino terminal amino acid is the valine. In case of alpha subunit, the carboxyl amino acid is arginine, whereas in case of beta subunit, the carboxyl amino acid is histidine. So when the hemoglobin, each the subunit has one heme pocket. That is, this heme pocket is present in the structure, and that's where the oxygen binding takes place. So this is the structure of the hemoglobin, and you can also see here clearly the each subunit has one heme pocket. That's where the oxygen binding takes place. And this is about the hemoglobin. And of course, the protoporphyrin ring systems are the tetrapyrrole prosthetic groups, not only present in the hemoglobin, they are also present in other cytochromes and other structures. And coming to the important function of the, what is the time? Yeah. Coming to the important function of the heme, why the heme is required the heme pocket is required for the O2 binding. In the sense, oxygen is not very highly soluble in water to transport in its free form, but in the human body, the hemoglobin transport its oxygen in the circulation from the lungs to the tissues, and no other amino acids, any amino acids can efficiently bind the oxygen except hemoglobin. And in hemoglobin also, the iron is highly sequestered highly sequestered, in the sense you can see here that the iron is not freely present. It is highly sequestered in between the pyrrole ring systems. So that means the free iron can generate the radicals, because Fe+2 is highly reactive. It can form many radicals through Fenton's chemistry as well as Haber-Weiss reaction, and this free iron can easily form hydroxyl radical, and this hydroxyl radical from the superoxide anion. And so that's why the iron in the human body is sequestered by the prosthetic heme prosthetic group. And this, when the iron is in a Fe+2 state in the prosthetic group, it is less reactive. In the sense, when it is present inside the heme pocket, it is less reactive. When it is present freely, it is highly reactive. That's why. And in addition to this, when it switches from Fe+2 to Fe+3 state, again it loses its O2 binding carrying capacity, and therefore this Fe+2 to Fe+3 conversion is also prevented by the 4 nitrogen atoms of the pyrrole ring system. So that's why totally the The free heme in free heme binding of oxygen can switch its oxidation state from Fe plus 2 to Fe plus 3. But in case of hemoglobin, since the iron Fe plus iron is sequestered by the 4 nitrogen atoms of the pyrrole ring, that conversion is prevented. So this is the function of the hemoglobin and its structure. And in addition to this, let me make two more points here. We will discuss the remaining point later. This hemoglobin is exists in two states. So one is oxy state, second one is the deoxy state. In oxy state, you can see here that it exists in two states. One is a deoxy state, another one is a oxy state. In a deoxy state, or it is also known as the deoxyhemoglobin, and it is also known as the T state or tensed state. Whereas when the O2 bound form of the hemoglobin is the R state, it is also known as the relaxed state or the oxyhemoglobin. So this is T state is nothing but the deoxyhemoglobin, whereas the R state is nothing but the oxyhemoglobin. So you can also see here, when upon binding of oxygen, the T state would be converted into the R state, or upon releasing of the oxygen from the oxyhemoglobin, R state would be converted into T. So that means these two states are interconvertible depending upon the condition and the oxygen tension. So you can, and the T state has it is a low affinity for the hemoglobin, whereas the R state has the high affinity for the hemoglobin. And the reason why we call T state deoxyhemoglobin is also called a T state or tensed state, in the sense, in deoxyhemoglobin, you can see here, as I already said, that hemoglobin is a tetramer, and it has 4 subunits, and the alpha 1, alpha 2, beta 1, and the beta 2. All these 4 subunits are highly stabilized by the the salt bridges. So that means in deoxyhemoglobin, all the 4 subunits are highly anchored by 8 salt bridges. You can see here 8 salt bridges are present. You can count here 1, 2, and 3, 4, 5, 6, 7, 8. That means 8 salt bridges are present when the hemoglobin is in the deoxygenated state. These 8 salt bridges are highly sequester the 4 chains, and they have no freedom for the rotation. So that's why it is also known as the tensed state or the taut state. And you can see here all these 8 salt bridges are present between the acidic amino acid and as well as the basic amino acid, or between the carboxylic or the amino group. So here aspartic acid, between aspartic acid and the histidine, and here the side chain amino group and the side chain carboxylic group, and so on. That means in deoxygenated state, the hemoglobin is highly anchored by 8 salt bridges. That's why all the subunits are highly anchored and they don't have the freedom to rotate, and that's why this is known as the taut state or the tensed state. And when upon oxygen binding, all these 8 salt bridges would be progressively disrupted. As a result, the T state becomes the R state. So this we will continue in the next class.